on nervous system physiology, and I'll begin here with neurons. So keep in mind, we're about to change resting membrane potential. So the changes in membrane potential, this is used as a signal, and the neurons use these changes in membrane potential to receive, integrate, and send information. So the two ways in which the changes in membrane potential occur are as follows. So one, you have some alteration in ion concentration on the two sides of the membrane, and two is anything that changes membrane permeability, of course, meaning changes the number of open channels. It will indeed change membrane permeability and, of course, transfers information. So two types of signals can cause these changes in membrane potential. So the two signals that are produced by this are graded potentials and action potentials. And I'll say that one more time. Those changes in membrane potential can produce two types of signals. The first of which is a graded potential. This is just an incoming signal that operates over a short distance, and that is opposed to the action potential. And the action potential class, it goes over a long distance. So it's a long distance signal of an axon. So with depolarization and hyperpolarization, they describe those changes in membrane potential relative to resting membrane potential. So depolarization is simply a decrease in membrane potential. So the intracellular side of the membrane becomes less negative, meaning it goes closer to zero. And if you haven't already, maybe you should draw the action potential from beginning to end. And then it's closer to zero, so it's less negative than the resting potential, which is about negative 40 millivolts to negative 90 millivolts. So this includes moving above that zero to become positive. So now you know what depolarization is. Let's move on to hyperpolarization. So hyperpolarization is an increase in membrane potential. So in this way, the intracellular side of the membrane becomes more negative, meaning going further from zero than the resting potential. For instance, maybe it goes from a negative 70 millivolts to a negative 65 millivolts. And if you're thinking in your brain right now which one that is, that would most certainly be depolarization. Yes, a very slight depolarization, but it is indeed. However, if I were to go from a negative 70 millivolts to a negative 75 millivolts, that indeed would be hyperpolarization, getting more and more negative. So now let's move on to the action potential. So keep in mind, that is the long distance signal. So the principal way that neurons send signals over long distances is by generating, yes, the first portion, and propagating or transmitting action potentials. So only cells with excitable membranes, known as A, neurons, and B, muscle cells, can generate action potentials. I'll say it again, only cells with excitable membranes, such as neurons and muscle cells, can generate action potentials. So the action potential is just this brief reverse, or this brief reversal of membrane potential with a total change in voltage, or a total change in amplitude of approximately 100 millivolts. So depolarization is followed by repolarization and a short hyperpolarization. And of course, this all occurs, class, in very short amount of time, meaning in milliseconds, unlike a graded potential. So unlike graded potentials, I say, action potentials, they do not decay with the distance. So they continue to go on and on and on and on, not decaying as a graded potential would do. So the nerve impulse, of course, is simply an action potential in a neuron and is typically only generated in axons. So, and it's only going to be generated when adequate stimulation is there. So the stimulus changes membrane permeability by opening specific voltage-gated channels. So from here, we're going to get into generation of an action potential. So we have four phases or four parts that are being included. So we have the resting state, which will be number 1. We have number 2, which is depolarization. 3 is repolarization. And then 4 is hyperpolarization. So let's begin the resting state. So at rest, all gated sodium and potassium channels are closed. So only leakage channels are open. to, of course, maintain that resting membrane potential. So each sodium channel has two gates. So there are those voltage-sensitive activation gates that are closed at rest periods. And of course, they then respond to depolarization by opening. And of course, in an activation gate, of course, it blocks a channel once it opens. From there, depolarization opens and then inactivates the sodium channels. So in contrast, each active potassium has a single voltage-sensitive gate, and that is closed during the resting state. It opens class very, very slowly in response to the depolarization events that occur. Let's get now to number 2, which is depolarization. With depolarization, the sodium channels open. So there's going to be local currents that depolarize the axon membrane, and then the voltage-gated sodium channels open, and sodium rushes into the cell. This influx of a positive charge depolarizes the local patch of the membrane further, opening more and more sodium channels. Hence, the interior of the cell becomes progressively less negative. When depolarization reaches that critical level, that threshold, which is approximately negative 55 millivolts to about negative 50 millivolts, depolarization becomes self-generating, meaning it's urged on by that positive feedback, going further and further depolarizing. So as more sodium enters, the membrane depolarizes further, opening more, and then of course finally all sodium channels. So the membrane becomes less and less negative, and then you get the overshoot that you love to draw each and every day, and that spike of the action potential. So membrane permeability and membrane potential, of course, they both cause positive feedback. Let's get on down to repolarization. So with repolarization, the sodium channels are inactivating and the potassium channels open. So it's the explosive action potential that lasts only about 1 millisecond. It is self-limiting because inactivation gates of the sodium channels close to inactivate. So membrane permeability to sodium declines, and then we get back to resting levels. So the action potential spike stops. The slow voltage-gated potassium channels open, and the potassium rushes out of the cell, following its electrochemical gradient, restoring internal negativity of the resting neuron. This is called repolarization, and it's caused by this abrupt decline in sodium permeability and this increased permeability to potassium that contributes to repolarization. Let's get to number 4 to finalize what's happening here. So now we're to hyperpolarization. So some potassium channels will remain open, and the same happens with some sodium channels. They reset. So we now have the period of increased potassium permeability, and it typically lasts longer than needed to restore the resting state. So excessive potassium efflux right before, and then the potassium channels close. So a hyperpolarization is seen with this slight dip after the spike, and then the sodium channels reset, reopening inactivation gates and closing their activation gates. So to end things, repolarization restores the resting electrical conditions, but not resting ionic conditions. So after repolarization, the sodium-potassium pump redistributes the ions. We've now made it down to threshold and the all or none phenomenon. So the depolarization must reach threshold values. And in case you've forgotten, it's approximately negative 55 millivolts to negative 50 millivolts. And it's, of course, that simply change of 15 to 20 millivolts from that resting membrane potential. So if an axon is to fire, it has to reach those threshold values. So the outward potassium movement is equal to the inward current of sodium movement. So if 1 more sodium enters class, depolarization occurs, opening more and more sodium channels. So if 1 or more potassium leaves, membrane potential is driven away from threshold, and of course those sodium channels close, and then more potassium channels open, getting us back to resting membrane potential. So an action potential is an all or none or all or nothing phenomenon, meaning it either happen or it does not happen. So let's get down to propagation of an action potential. So, an action potential class must be propagated along the axon's entire length, meaning both those that are non and those that are myelinated. So, with this happening, what we have now, class, is that the action potential has to be generated by an influx of sodium through a given area of the membrane, and as that happens, it begins to establish the local currents that will depolarize adjacent membrane areas in the forward direction away from the origin of the nerve impulse, which of course opens voltage-gated channels and triggers an action potential there. So if in fact, class, it's a non-myelinated neuron, of course it's going to be because the area where the action potential originated and just generated an action potential, the sodium channels in that area are inactivated, meaning no new action potential generated there. Hence, the action potential propagates away from its point of origin. So the action potential initiated at one end of an axon and conducted away from that point toward the axon's terminal. So once initiated, action potentials are self-propagating and continues along the axon at a constant velocity. And of course here it's slow and it just happens as dominoes would on a table falling in sequence. So the presence of a myelin sheath dramatically increases the rate of action potential propagation. So let's find out what happens when things are myelinated, you all. So myelin, in case you've forgotten, is an insulator. It prevents almost all of the charge from leaking from the axon and allows the membrane voltage to change more rapidly. So the current, yes, I said the current can only pass through the membrane of a myelinated axon at myelin sheath gaps we refer to as nodes around VA. Those nodes around VA are where there is no myelin and the axon is bare. So nearly all voltage-gated sodium channels are concentrated in these gaps. And in case you're wondering, those gaps are about 1 millimeter in length. So the action potential that's generated, meaning action potential generated, the current moves rapidly to the next node around VA, where it triggers another one. So this movement, class, from node to node to node is referred to as saltatory conduction. So not dissipating through adjacent membrane areas, as it happened with those non-myelinated axons. So these electrical signals jump from node to node 30 times faster than those that are non-myelinated. Now let's get to those refractory periods. So there are two types, the absolute refractory period and the relative refractory period. The absolute refractory period is from the opened sodium channels until those sodium channels begin to reset to their original resting state. So it ensures each action potential is a separate, all or none, one-way transmission. The relative refractory period follows the absolute refractory period. So most sodium channels are in the resting state and some potassium channels still open for repolarization that's occurring. Threshold for an action potential class is substantially elevated, so a stimulus that would normally generate an action potential is no longer sufficient during this relative refractory period. Now let's go to synapses and the way synapses transmit signals between neurons. So we have the chemical synapse. So at the chemical synapse, of course, an action potential arrives at the axon terminal, beginning with neurotransmission. And of course, this will be at the presynaptic axon terminal. That's number one. 2, the voltage-gated calcium channels open, and of course, calcium enters the axon terminal, giving us depolarization that opens sodium channels, class, and those voltage-gated calcium channels. So the calcium channels flood. The calcium channels allow calcium to flood down the electrochemical gradient from extracellular areas to the intracellular areas into the axon terminal. So the calcium entry causes those synaptic vesicles to release neurotransmitters via exocytosis, giving us this surge. So this surge of calcium into the axon terminal acts as an intracellular messenger. And then the synaptic vesicles fuse with the axon membrane and empty contents via exocytosis into the synaptic cleft. So calcium is quickly removed from the terminal and is taken up via the mitochondria, or at least by mitochondria, and ejected from the neuron class via the calcium pump. At number 4, the neurotransmitter diffuses across the synaptic cleft and binds to specific receptors on the postsynaptic membrane. 5. Binding of the neurotransmitter class opens ion channels, creating greater potentials. Once the neurotransmitter binds to receptor proteins, the receptor changes shape, opening ion channels, creating greater potentials. So the postsynaptic membranes contain chemically gated ion channels, and of course are packaged as receptor proteins and ion channels. So depending on the receptor protein to which the neurotransmitter binds, the postsynaptic neuron may be either excited or inhibited. Depending on the postsynaptic neuron, the neuron, of course, may be either excited or inhibited. So, number 6, the neurotransmitter effects are terminated. So when you have binding of the neurotransmitter to its receptor is reversible as long as the neurotransmitter is bound to the postsynaptic receptor. A neurotransmitter continues to affect membrane permeability and block reception of additional signals from the presynaptic neurons. Hence, those effects typically last a few milliseconds before being terminated. So this happens in one of 3 ways. So it would be via reuptake, degradation, or diffusion. So if it's by reuptake, of course, the astrocytes or the presynaptic terminal is where it's stored, of course, or where it's destroyed. If it's via, and the one example that's reuptake in class is norepinephrine. If it's going to be degraded, of course, it's via enzymes associated with the postsynaptic membrane and the synaptic cleft. In that case, you know about acetylcholine. And then finally, class, it could very well just diffuse away from the synapse. This now completes threshold potential, including the all or none phenomenon, even to propagation of an action potential via those roads of Rambier, onto refractory periods, and then finally, class, the synapse. So from now, I would say study well and even practice writing, because on the test you will be expected to explain the events occurring during a nerve impulse. If you need any help, let me know. Study well, class, and I'll see you later.